Imagine building with Lego spot on a molecular level. That's kind of what supramolecular plastics are all about. They're made by carefully arranging molecules so they stick together through weak, reversible bonds. This gives them unique properties. Many people confuse organic chemistry with supramolecular chemistry. So, let's break that down. Organic chemistry concentrates on studying the covalent bonds, which means it works with perhaps six different atoms and the scope of interactions is quite limited. But supramolecular chemistry deals with another kind of scale. It deals with the interactions between all molecules, which are weaker, but explores the interactions between molecules and their organization into larger structures through non-covalent interactions. Japanese researchers synthesize different monomers of guanidinium sulfate base. To be more precise, here are the schemes. 1,7-diaminoheptane was added to an aqueous solution of methylazoate hemisulfate salt. All the next steps were described detailed in the article. The resultant solid residue was dried, affording white powder. To an aqueous solution of cysteamine dihydrochloride was added potassium hydroxide. Subsequently, an aliquot of cysteamine thus obtained was immediately added to an aqueous solution of O-methylazoate hemisulfate salt. The resulting solid residue was dried as well, affording white powder. Monomer amine guanidinium was synthesized according to a procedure similar to that used for alkyl-guanidinium. This 2-aminoethyl ethane 1,2-diamine was added to an aqueous solution of O-methylazoate hemisulfate. A crystalline substance was isolated and dried in a vacuum over 120 degrees Celsius for 12 hours, affording white powder. After successfully synthesizing these, they synthesized supramolecular polymers by adding an aqueous solution of sodium hexametaphosphate, whereupon the mixture was immediately emulsified. A condensed microscopic phase formed via centrifugation was isolated and washed 3 times with water. The resultant viscous liquid was dried in a vacuum oven at 95 degrees Celsius for 30 minutes, yielding alkyl supramolecular polymer in the form of colorless glassy solid. All other supramolecular polymers were likewise obtained. Right now on the slide you can see selected snapshots of the spontaneous liquid-liquid phase separation event upon mixing aqueous solution of guanidine monomer and sodium hexametaphosphate immediately after mixing. After these steps, they added an aqueous solution of chondroitin sulfate to an aqueous solutions of the monomers. They performed 3D printing of guanidine sulfate supramolecular polymers on a slide glass at 30 degrees Celsius by using a sorbate from guanidine sulfate and amine guanidine. So, except of all that, Japanese conducted a brilliant research in general. They used a whole toolbox of scientific methods, such as seeing the invisible. Well, they used a method that studies how matter interacts with light, spectroscopy. But let me explain how does it work. When an atom is hit with light quanta or a photon, the energy of that photon is absorbed by an electron, which then moves to higher, more energetic orbital. The amount of energy needs to be exactly the same as the amount that an electron needs to move to higher orbital. So not all photons will be absorbed by all atoms, nor do all atoms emit the same photons, right? Different atoms have different number and arrangement of electrons, which is crucial. And that arrangement determines how electrons can move between orbitals. This means that different elements will all be able to absorb and emit photons of different energy and thus of different color as well. So that's why they use this method to identify the building blocks and functional groups within the plastics, like a chemist's fingerprinting kit. So for unraveling structures, they used X-ray diffraction methods, which works by directing a beam of X-rays at a crystalline sample and analyzing the way the X-rays are scattered. The scattered X-rays create a diffraction pattern that reveals information about the arrangement of atoms within the crystal. This technique is used to determine the crystal structure, chemical composition, and physical properties of materials in general as well. So that's how it helped them to see the internal architecture of these plastics. So differential scanning calorimetry allow them to see how the plastics behave when heated by measuring the difference in heat flow between a sample and a reference material as they are subjected to a controlled temperature program. Reshaped supramolecular polymers, researchers first partially dried them under milled vacuum overnight. These made them soft enough to mold into shapes like a heart before fully curing. For dumbbell shapes, supramolecular polymers were hot pressed at a temperature 20 degrees Celsius above their glass transition temperature to form thin films. These films were then cut with a dumbbell-shaped cutter on a hot plate. Also, they conducted an adhesion durability test. They glued two glass plates together using a sticky, oil-like supramolecular polymer. A droplet was placed between the plates, and a 500-gram weight was used to press them together. In underwater and artificial seawater conditions, a lighter 350 grams weight was used. Purpose: this checks how well the material sticks over time, even in water. Also, they conducted thermal durability tests. Small pieces of supramolecular polymers film were placed in a 100 degrees Celsius oven for up to 14 days. After that, the material surface was tested for hardness and indentations. Purpose: they showed whether the material stays strong after long exposure to high heat. And also water durability tests. A large piece of supramolecular polymer film was soaked in water with 500-gram weight placed on top. Purpose: this checks if the material can resist breaking down or deforming in water over time. But that's not all. They also performed tensile tests to measure just how strong these materials are, seeing how much they could stretch before breaking. They also used dynamic mechanical analysis to understand how the plastic respond to stress over time and at different temperatures, like seeing how rubber band behaves when they stretch it repeatedly. The same thing, actually. However, the most significant feature about this plastic is its biocompatibility and non-toxicity to the environment. To prove this, Takuzo Aida and his team conducted AIMS tests. The main essence is here. Method uses bacteria to test whether a given chemical can cause mutations in the DNA of the test organism. More formally, it is biological assay to assess the mutagenic potential of chemical compounds. So, for Salmonella typhorum strains and one Escherichia coli strain were incubated with supramolecular polymers in the presence and absence of an exogenous metabolic activation system from mice liver at different concentrations along with appropriate positive and negative controls. The assessment of the results was based on the number of relevant colonies in each plate, and the results were regarded as mutagenic when the number of reverting colonies was more than twofold of the negative control. Results ensured the materials weren't harmful and any potential mutations in bacteria were observed. But salts are also present in soil in quite big quantities. That's why they tested their material on soil to see how they naturally break down over time. And it showed promising results. Decomposition takes 200 hours. And that's how they showed how these materials could be broken down and reformed, demonstrating a truly circular economy for plastics. So, to conclude, a revolutionary type of plastic called alkyl supramolecular polymer has showed the best results. It was developed by scientists from the Riken Institute in a collaboration with the University of Tokyo. This material has high strength, comparable to traditional petroleum-based plastics, can withstand heating up to 120 degrees Celsius, and can be used to produce packaging, films, and solid products. The unique feature of this plastic is that under the influence of salt, it quickly breaks down into its original monomers. In seawater, this process takes only 1 to 3 hours, while in saline soil, the material fully decomposes in approximately 8 days. The dissolution happens because the salt water disrupts the chemical bonds between the two monomers, after which natural bacteria complete the decomposition process. As a result, the plastic does not leave behind harmful microplastics, does not release carbon dioxide, and turns into substances that are harmless to the environment. According to the developers, the material is non-toxic and non-flammable. Thanks to its use of supramolecular compounds, it opens up wide possibilities for the packaging industry. This technology is expected to significantly reduce global ocean pollution, protect marine life, and lower risks of microplastics entering the food chain. The United Nations Environment Program predicts that plastic pollution will triple by 2040, adding 23 to 37 million tons of waste to the world's oceans annually. Despite this disturbing news, I want to end this video with a quote from the head of this dazzling study. Children do not get to choose the planet they will live on. It is our duty as scientists to leave them the best possible environment, said Aida. So here are the links I used creating this presentation. You will be able to find them in the description. And on this moment, I want to thank you for your time and remember, keep learning.